हाँ, chat box. Uh, okay. Hi everybody, welcome back to PW Gulf. I'm Jyoti, your zoology faculty, and yes, today we will be talking about one more new topic, which is going to be small because we have to read the NCRT part. Okay. Before we get started, please tell me if I'm audible and visible to you. Am I audible, everybody? And visible?
Yes, ma'am.
Yes.
Yes, ma'am.
Okay, great. Please, everybody, switch on your camera. I can only see Adina and Sufi. Aditri is also here. Where is Shafeen? Shafeen, what about you? Eliza, switch on your camera. Abdul, switch on your camera. Zuha, switch on your camera. I have got your mother's permission, right? You do not have any issue with the camera. Switch on your camera, Zuha. Mariam, switch on your camera. Pankti, what about you? Switch on your camera. Shafiq, switch on your camera. Everybody switch on your camera. Tell me how was the day, if you have any doubts from the last class. Oh, okay.
No, ma'am, and the day was good.
Okay, nice, nice. What about the topics? Are we all clear about the topics that we have learned in the last class? Yes, okay.
Yes, ma'am.
Okay, all right.
The difference between recognition, restriction site and recognition sequence.
Both are the same thing. Recognition site, recognition sequence, restriction site, same thing. Okay? Yeah. Like different names for the same thing. These are the different name for the same thing. Oh, hello, Pankti. How are you doing? It's been a long time, I guess. Hi. Sriniti, please switch on your camera. Everybody, please switch on your camera. So, today it will be a reading session. We have NCRT dialogues and you have to read those part so that we are clear about all the things. Can we play Kahoot again? Abdul, we will play, but not today. Okay. Once we cover up, good amount of section from your syllabus, then we will have the Kahoot quiz again. Okay, I know you missed it, but yeah, don't worry. Okay, you want the same Kahoot quiz, but it is no fun. Everybody knows the answer. Right. So, Abdul, we will have a Kahoot quiz maybe in the next class. Okay. Okay.
Okay, we will prepare then.
Yes,
Tell
be
me what
prepared.
topic.
Be prepared, everybody. Okay. So, I think without any further ado, I think we can start with the reading so that you guys get free soon. Let's talk about this part. Cloning vectors we were talking about. So, this part is left from NCRT. Who is going to read? Should I choose you randomly? Let's start with Shafiq. Okay, Shafiq, if you can see this paragraph, you can read this. Yeah.
The screen is black. Something is really close. Okay.
Okay.
You know that plant and bacterial phages have the ability to replicate within bacterial cells independent of the control of chromosomal DNA. Bacterial phages, because of their high number per cell, have very high copy numbers of the genome within the bacterial cell. Some plasmids may have only one or two copies per cell, whereas others may have 15 to 100 copies per cell. Their numbers can go even higher. We are able to link an alien piece of DNA with bacterial cells or plasmid DNA. We can multiply its numbers equal to the copy number of the plasmid or bacterial cells. Vectors used at present are made in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants.
Okay. Nice. So everybody, you know that we have plenty of cloning vectors that we can use. Some of the examples of cloning vector, we have plasmids like PBR322, we have plasmids such as PUC19, we have plasmids such as TI plasmid of your Agrobacterium tumefacium, right? So there are different type of vectors. We have bacteriophages, we have plasmids which can, once they enter inside the host cell, they can just multiply independent of the host cell. Okay, they just have to enter inside the host cell, and then using the DNA machinery of the host cell, like the DNA polymerase enzyme and the proteins, they will be able to replicate without any interference from the host cell. Okay, so what do we have? We have bacteriophage, we have plasmids, and these plasmids and bacteriophage, they can replicate inside the host cell like a bacteria or any other cells. Okay, so plasmids and bacteriophages, they have the ability to replicate within your bacterial cells, which is independent of the host cell. Now, important thing is, bacteriophages, we all know, viruses, they can easily just multiply, replicate, make their multiple copies within the host cell. For example, HIV virus. HIV virus, once it enters inside your macrophage becomes the factory of HIV virus, right? Because they are able to make their own multiple copies. They can make n number of copies within the macrophage, and those clones of HIV virus, they can be released and they can infect the T helper cells. Okay. So, talking about bacteriophage, they have high copy number. They can easily just make multiple copies within the host cell. But if you talk about plasmids, they can have variability in copy number. Some of them, they can have copy number of 1 or 2. So only 1 copy of plasmid, only 2 copy of plasmid will be formed within the host cell. But some of the plasmids, they are having high copy number such as 15 to 100 of copy number. So within one single cell, plasmids will be able to make hundreds of copies of themselves within the host cell. Okay, and along with the plasmids, of course, your gene of interest is ligated in the plasmids. So as the plasmids are replicated, your gene of interest will also be replicated. Okay, so simple thing is that just remember that plasmids, they can have different copy numbers. Some of them, they can have 1 to 2 copy number, which is very low. Okay, and some of them, they can have copy number which can be 15 to 100. Okay, and whatever copy number is there for the plasmid, that many number of gene of interest copies will be formed. Okay, for example, if we have a plasmid, and to this plasmid you have ligated your gene of interest, this plasmid is having a copy number of, let's say, 30. Okay, so of course, after entering inside the host cell, this is going to make 30 copies of plasmids of itself. Okay, and along with 30 copies of plasmids, 30 copies of our gene of interest will also be formed. So it depends how many copies do we want. Okay, if I want so many number of copies of my gene of interest, so I'll be using a vector which is having high copy number. Simple thing, right? So yes, if you talk about the plasmids, they can have some plasmids, they can have 1 or 2 copy number per cell, and if you talk about some other plasmids, they can have high copy number which can be around 15 to 100 copies per cell. Okay. Now, their number can go even higher if we are able to link an alien piece of DNA with bacteriophage or plasmid DNA. We can multiply its number equal to the copy number. So, the same thing. If you are able to link an alien piece of DNA, a gene of interest, with your plasmids near the ori region, of course, whatever copy number your plasmid is having, suppose 30 copy number your plasmid is having, so that many number of copies of your gene of interest, of your alien DNA will also be formed. Okay, vectors that we are using at present, of course, these are not your normal vectors, natural vectors. These vectors, they are prepared in a lab in such a way that they do not have any non-useful genes in them. They have the least size as possible so that they can be transferred easily. Also, easy linkage of our gene of interest can happen. Okay, so yes, vectors that we are using at present, they are engineered in such a way that help us to easy linking of foreign DNA. And how we are able to link a foreign DNA? We are able to link a foreign DNA because when we are engineering your vectors in the lab, we are making sure that they have recognition site, they have restriction site for particular restriction enzyme. So that whenever we want to link an alien piece of DNA, we just have to use a restriction enzyme, make a cut, and gene of interest will be ligated. Okay, so easy linkage of foreign DNA is possible because of the recognition site. Okay, recognition site, or you can call it as cloning site. Okay, that can be present within your vector. Okay, then selection of recombinant from the non-recombinant. Okay, so yes, remember this thing, we are able to easily see which plasmids are recombinant and which plasmids are non-recombinant. The recombinant plasmids, of course, they have lose their ability to grow on tetracycline antibiotic, or they have lose their ability to produce a color. Okay, so very easily we can choose recombinant from the non-recombinant if your vector is having what? If your vector is having selectable marker, right? So this is one thing. Now let's talk about this paragraph. So who is going to read this paragraph? Adina, maybe you can read. Okay, let me just get a side.
Sure, ma'am. Yeah, great. The following are the features that are required to facilitate cloning into a vector. Origin of replication ORI. This is a sequence from where replication starts, and any piece of DNA when linked to this sequence can be made to replicate within the host cell. This sequence is also responsible for controlling the copy number of the linked DNA. So if one wants to recover many copies of the target DNA, it should be cloned into a vector whose origin supports high copy number.
Nice.
Selectable marker. In addition to that,
No. If you want to read, you can go ahead. Yeah, go ahead. Yeah.
selectable marker. In addition to ORI, the vector requires selectable marker, which helps in identifying the eliminating non transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. You will study this process in subsequent sections. Normally, the genes including resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin, etc., are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against these antibiotics.
Okay, nice. So everybody, now let's talk about the features that we want in our vector to be there. Okay, so that easy cloning can happen. Successful clones can be formed. Alright, so first of all, the number one feature that we want to be there, without which of course the cloning will not be possible, is the ORI region. If ORI region is not there in your vector, then the replication of our gene of interest will not take place. Whatever we want, whatever product we want to be produced, that product will not be formed. So ORI region is the most important feature. ORI region should be there. If ORI region is not there, even if you are able to link your gene of interest, even if you are able to transfer this recombinant plasmid into the host, your gene of interest will stay like that, and none of its copy will be formed, and none of its copy will be transferred into the daughter cells. Okay, the new cells which are going to be formed, they will not have your gene of interest. So first of all, to have successful cloning of gene of interest, we want to have this ORI region. And in the ORI region, what do we have? We have the coding sequence from which the replication of the DNA, the plasmid, is going to start. And near this ORI region, we are going to ligate, we are going to insert, we are going to attach our gene of interest. I am not saying that just near this ORI region, just at the ORI region, you have to ligate your gene of interest, but it should be near to it. Okay, if you have a plasmid and if you have ORI region here, maybe somewhat here you can ligate your gene of interest, okay, so that when replication process start, your gene of interest is also replicated, okay. Okay, so yeah, in the ORI region, what do we have? We have the coding sequence from where the replication starts and the piece of DNA is replicated. The piece of DNA is going to be multiplied, okay. And one more thing that you need to remember, this sequence, it is also responsible for controlling the copy number of your linked DNA. So if you want a high copy of your linked DNA, if you want multiple copies of your gene of interest, you need to choose a vector whose ORI region supports high copy number. Okay, so remember this thing. ORI region, coding sequence from which the replication process begin, and this ORI region is going to support high copy number depending upon the vector in which it is present. Okay, so that is important. Now, talking about selectable marker, you already know selectable markers. These are the genes which are coding for antibiotic resistance gene, antibiotic resistance property, or they are coding for reporter enzyme, and they are helping us to choose transformant from the non-transformant, or it would be better to say recombinant from the non-recombinant. Okay, to make you understand. Yes. Let's have the explanation again. Okay. Different cases can be there. We have 3 sets of bacteria. Suppose we have plasmid. Okay. We have one more plasmids. Different type of plasmids are there. Some plasmids are there who are not having our gene of interest, but some plasmids are there which are having gene of interest. Okay, now depending upon 3 cases, we can have transformant and the non-transformant. Okay, so case 1, where the bacteria is having its chromosomal DNA, it is having its own plasmids. Okay, it is having its own plasmids, but none of the plasmid is absorbed by this bacteria. Okay, it will be called as non-transformant. Okay, then we have this plasmid which is called as non-recombinant plasmid. This non-recombinant plasmid, it is going into your bacterial cell. Okay, this bacterial cell is having its own chromosomal DNA, some of its plasmids, and it is absorbing this non-recombinant plasmid also. Okay, so I have told you technically this will be called as transformant. Okay, but it will be a recombinant or a non-recombinant transformant. It will be a non-recombinant plasmid because the gene of interest is not there. Okay, so yes, just remember that we are having a transformant here. With non-recombinant plasmid. Okay, or maybe you can just simply write non-recombinant transformant we are having. Okay, then another case we are having where we have a recombinant plasmid, and this recombinant plasmid is absorbed by your normal bacteria, E. coli. We are having its chromosomal DNA, we are having its own plasmid, and then it is also absorbing a recombinant plasmid with the gene of interest. So this one will also be called as transformant, but this transformant is having the recombinant plasmid. Okay. Are we clear about this part? I think they have removed the non-transformant part from the NCRT, because today only when I was taking screenshot from the updated NCRT, they have not mentioned non-transformant. Okay. But in the class, I told you that even though this one is a transformant cell, in the NCRT, they have mentioned non-transformant, right? So please make a correction in your previous notes. Okay. The cell which will grow on both the antibiotic, it will be called as non-recombinant transformant or a transformant with non-recombinant plasmid. Okay? Yes. Wherever you have written non-transformant, just correct that part and please write it will be a transformant but with a non-recombinant plasmid. Okay? Okay, Abdul. Got it. Yes, yes, Shafeen. It was like, in the NCRT it was written as non-transformant, but now it is not there in the NCRT. I guess I have a very old NCRT. So, yeah, but I always told you that, yeah, even if a bacteria is taking up any kind of foreign DNA, we will call it as transformant only. Okay, but in NCRT it is non-transformant. But just remember, you correct wherever you have written, guys, wherever you have written, just correct that part, that we will call it as a non-recombinant transformant or a transformant with a non-recombinant plasmid. Okay, that would be better. That would make more sense to you. Okay. I hope, Abdul, you have corrected this part. Okay, before going, just make sure you correct this part, otherwise you will forget. Everybody else, please make sure if you have made notes and in the classes, we have talked about transformant and a non-transformant. Remember, both the cells will be transformed, but one with the recombinant plasmid and the one with non-recombinant plasmid. Okay? Clear? Everybody, is it clear? I'm going to ask you in the end of the class. Okay? Maybe any one of you randomly I'll ask whether the cell is transformed or not. Okay? If this part is clear to everybody. So this is case 1 where the cell is not having any foreign cell, any foreign DNA molecule, any foreign DNA piece. So it will be called as non-transformant. But here it is receiving a non-recombinant plasmid. Okay, it is foreign to the cell. So of course it will be a transformant. Here this recombinant plasmid is introduced into the cell. So again it is transformant. But the difference is this transformant is without a recombinant plasmid. It is having a non-recombinant plasmid. Okay, and here we are having a recombinant plasmid with the transformant. But both of them, they are transformed. Okay, I think it is clear. So moving back, everybody, selectable marker, we talked about this part. Selectable markers, what are these? These are basically the genes which are coding for resistance to antibiotic such as ampicillin, chloramphenicol, tetracycline, kanamycin. All these are antibiotic. And if we have antibiotic resistance gene, the cell is also going to have the antibiotic resistance property. Okay. And on the basis which cell is able to grow on one antibiotic and which cell is able to grow on both the antibiotic, we can easily choose a transformant, okay, from the other transformant which are having non-recombinant plasmid. Okay, so we have transformants, but we are choosing a transformant with a recombinant plasmid. Okay, yeah. Okay, if this is clear, then let's move ahead and let's talk about this part. So who is going to read the cloning site? Cloning site. Hmm.
Ma'am, can I go?
Yes, yes, Sumaya, go ahead. Please read.
Cloning sites. In order to link the alien DNA, the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzymes. Presence of more than one recognition site within the vector will generate several fragments which will complicate the gene cloning. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. For example, you can ligate the foreign DNA at the BAMH1 site of tetracycline resistance gene in the vector PBR322. The recombinant plasmids will lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant ones by plating the transformants on ampicillin containing medium. The transformants growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinants will grow in ampicillin containing medium but not on that containing tetracycline. But non-recombinants will grow on the medium containing both the antibiotics. In this case, one antibiotic resistance gene helps in selecting the transformants, where the other antibiotic resistance.
Okay. Okay, we can read. Yeah, you can read till this part. Okay.
Antibiotic resistant gene gets inactivated due to insertion of alien DNA and helps in selection of recombinants.
Okay. Good. So yes, third feature that we want in our cloning vector is the cloning site. And this cloning site is nothing but the recognition site, or you can call it as restriction site for the restriction enzyme. Okay. And how many restriction site do we want? We want a single restriction site, a single recognition site, or a single cloning site for commonly available restriction enzyme. If you talk about EcoR1, only a single restriction site is present. If you talk about SAL1, a single restriction site is present. BAMH1, a single restriction site is present. Okay, so we want cloning site. A cloning site is basically the recognition site where restriction enzyme is going to make a cut, and at that cut, we are going to have sticky ends, with the help of which, of course, the gene of interest will be ligated there, right? So in order to link alien DNA, we want a recognition site for the restriction enzyme. Okay, it has to have either very few, but we want to be a single site so that only a single cut is there and gene of interest can be easily ligated there. Okay, so preferably we want a single recognition sites for commonly used restriction enzyme, so that we have multiple option of restriction enzyme that can be used. But of course, all those restriction enzymes, they are going to just have a single cloning site, a single restriction site in the vector. Okay, presence of more than one recognition site within the vector, it is going to create problem, because this will generate, if we have more than one recognition site, it is going to generate multiple fragments, which will complicate the whole gene cloning process. Multiple fragments are there, we will have to assemble it, and then we have to assemble, attach the gene of interest, so it is going to be a difficult task. Also, if we have so many restriction site, it is going to make multiple cuts in the plasmid, due to which plasmid molecule will also be destroyed. Okay, so the ligation of alien DNA is carried out at the restriction site, and this restriction site should be present if you are using antibiotic resistance gene as the selectable marker. So it should be present in either of the one selectable marker which is there. Now, question in your NEET exams, they can come like this. They can just simply ask you this whole diagram. Everybody, you should be clear with this diagram. Okay, they will not give you diagram in the question, but they can ask you if you want to make a cut at ampicillin resistance gene, which of the following restriction enzyme you are going to use. Okay, so you know, if you want to make a cut at ampicillin resistance gene, you have two options. Okay, and these two options should be present in those given options. Okay, so you have to choose either PST1 or PVU1, right? These are the enzymes which you can use to make the cut. Similarly, if they are asking you which of the following enzyme has the restriction site as at the BAMH1, which of the following restriction enzyme they have the site at tetracycline resistance gene. So of course the answer is going to be BAMH1. Okay, So remember this thing. They can ask you anything. At this particular gene, which enzyme can be used? Which enzyme is having the recognition site? Okay. Which enzyme can be used to clone the gene of interest? So that will be BAM H1 in that case. So remember the whole diagram in the correct order. Okay. For example, they can ask you which of the following enzymes they have the restriction site at the same region. So these three restriction enzyme, they have the cloning site, they have the recognition site at the same region. Okay, in this particular blue coding sequence region. Okay, so these types of question can come. Okay, if we are clear about this part, everybody, we have learned about how do we choose a transformant from the other transformant who are having non-recombinant plasmid. Okay, so to choose the transformant with recombinant plasmid, of course, we are going to ligate our gene of interest um, at BAM-H1 site of tetracycline resistance gene of PBR 322 vector, due to which the recombinant plasmid, yeah, the recombinant plasmid, it will lose tetracycline resistance, okay, and we can easily choose recombinant from the non-recombinant, or you can say transformant having the recombinant plasmid from the transformant having non-recombinant plasmid, okay. So yeah, here we are able to choose recombinant from non-recombinant. The transformants which are there, all of them who have received the alien DNA, they will be called as transformant, okay. They will be cultured on ampicillin containing medium, and then they will be cultured, they will be transferred on a medium containing tetracycline. The recombinant will grow only on ampicillin medium and they will not grow on the tetracycline medium. Okay, but if you talk about the non-recombinants, okay, plasmids, transformants which are having non-recombinant plasmids, they will be able to grow on both the medium. Okay, so yeah, non-recombinant, it will grow on medium containing both the antibiotic. And in this case, one antibiotic resistance gene is destroyed because of the ligation of our gene of interest on the tetracycline resistance gene. So it will be able to grow on ampicillin only, and that particular clone, we can call it as recombinant or we can call it as transformant with the recombinant plasmid. Okay. So yeah, that is the thing. In this case, one antibiotic resistance gene helps in selecting the transformant, whereas other antibiotic resistance gene will be inactivated due to the insertion of alien DNA. So that's why we want at least two antibiotic resistance gene. So that one will be inactivated and it will result in the clones that will grow on one antibiotic only, and some clones grow on both the antibiotic. So we will easily choose our transformant with the recombinant plasmid. Okay, if we are clear about this part, next person who is going to read? Yes, raise your hand if you want to read, or should I choose the people, students randomly? Choose. Farhan, go ahead. Ma'am, inky, pinky, ponky, what do you mean, Sufi? Oh, wait, 1 second, Farhan. Yeah, what was that, Sufi?
Ma'am, when you don't know what to choose, you just go like inky, pinky, ponky, and then choose that person.
Oh, okay, okay, got it. Well, Farhan, you go ahead. You read this paragraph.
Yeah, I got disconnected now. So
Yeah.
gene gets inactivated due to insertion of alien DNA and helps in selection. Oh, am I supposed to read that one? The one on the left, right?
Yeah. This one. Yeah.
Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating of two dates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substance. In this, a recombinant DNA is inserted within the coding sequence of an enzyme, beta-galactosidase. This results into inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. The presence of chromogenic substrate gives blue-colored colonies if the plasmid in the bacteria does not have an insert. Presence of insert results into insertional inactivation of the beta-galactosidase gene, and the colonies do not produce any color. These are identified as recombinant colonies.
Okay, nice. Okay, so now everybody, we learned about that whenever we are using the gene which are coding for antibiotic resistance, there we have to have two antibiotic resistance gene, and that procedure require 2 days because we have to do plating on one antibiotic on one day and on the other antibiotic on the other day, right? So that makes it a difficult task. We have to have two identical setup. Also, it takes 2 days to proceed with the procedure. So instead of that, what we can do, we can have one alternate selectable marker which is present in your PUC 19 vector. Okay, this selectable marker, it is actually a gene which is coding for beta galactosidase enzyme. Okay, so remember this thing, an alternate selectable marker is developed which differentiate your recombinant from the non-recombinants on the basis of their ability to produce the color in the presence of chromogenic substrate. So please remember, it is not substance but substrate. Okay, you have to use the word substrate. So basically we are using the selectable marker where we are able to just choose recombinant from the non-recombinant on the basis of their ability to produce color in the presence of a chromogenic substrate, for example, X-gap. In this, a recombinant DNA, it is inserted within the coding sequence of the gene which is coding for beta galactosidase enzyme. We very well know that, okay, we are going to have a plasmid, and that plasmid is actually having a gene which is called as LAC-Z gene. Okay, so yes, what do we have? We have LAC-Z gene, and in the coding sequence of this LAC-Z gene, we are inserting our gene of interest. Okay, so gene of interest is ligated within the coding sequence of this LAC-Z gene, due to which beta galactosidase enzyme will not be formed, and this beta galactosidase enzyme, it can produce blue color if the chromogenic substrate is there, and if it is inactivated, no blue color will be produced. So just remember, DNA is inserted within the coding sequence of an enzyme beta galactosidase. Due to this, insertional inactivation will take place for that gene. Due to this, chromogenic substrate which is producing blue colored colonies, if the plasmid of bacteria does not have an insert, if the plasmid of the bacteria is a non-recombinant, the gene is activated, the gene produces enzyme produces blue color because of the chromogenic substrate. But if we have insertional inactivation, gene will be inactivated, enzyme will not be formed, no blue color will be produced because of chromogenic substrate. Okay, so yeah, we are able to choose recombinant from non-recombinant if they are just growing in transparent color, and some of them, if they are growing in blue color, they will be called as transformant with the non-recombinant plasmid. Okay, I think guys, to just clear this confusion that I have, I think you guys be confused with transformant and non-transformant. You can just write, instead of transformant and non-transformant, you can just simply write recombinant and non-recombinant. Okay, instead of transformant and non-transformant, just write recombinants and non-recombinants. Okay, that would be much better. Just replace transformant with recombinant, non-transformant with the non-recombinant. That would be much better, because in NCERT they have not talked about non-transformant. So yeah, that would be creating confusion. Okay, if we are clear about this part, now let's talk about the examples of vectors which are used for cloning vector. So the next person who would like to read, or should I go with Inky Pinky Ponky? Well, chalo. Let me ask. Let me ask. It's so difficult to choose you guys. Like, yeah, Sufi, Sufi, you go ahead. Yes. This paragraph for you.
Okay. Vectors for cloning genes in plants and animals. You may be surprised to know that we have learned the lesson of transferring genes into plants and animals from bacteria and viruses, which have known for which have known this for ages. How to deliver genes to transform eukaryotes is one. For example, Agrobacterium tumefaciens,
Yes, yes.
pathogen of several. Oh, 1 second, the screen blurry. Okay. A pathogen of several dicot plants is able to deliver a piece of DNA known as pDNA to transform normal plant cells into a tumor and direct these tumor cells to produce the chemicals required by the pathogen. Similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. A better understanding of the art of delivering genes by pathogens in the eukaryotic host has generated knowledge to transform these tools of pathogens into useful vectors for delivering genes of interest to humans. The tumor-inducing plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector, which is no more pathogenic to the plants but is still able to use the mechanisms to deliver genes of our interest into a variety of plants. Similarly, retroviruses have also been disarmed and now are used to deliver desirable genes into animal cells. So once a gene or a DNA fragment has been ligated into suitable vector, it is transferred into a bacterial, plant, or animal host, where it multiplies.
Nice. Nice. So, we know there are plenty of vectors that we can use. Even though this knowledge of biotechnology technique is kind of new for the humankind, there were many pathogens which were using this technique of biotechnology, that is to insert the gene of interest into the host cell so that they can achieve whatever product they want. Okay, it is very common. It has been known for ages by these pathogens like some bacteria and the viruses. Okay, so remember this thing. You would be surprised to know that we have learned the lesson of biotechnology. We have learned about some techniques of biotechnology. These lessons we have learned actually from the pathogens. Okay, pathogens, and these pathogens, they are like very pro with how to deliver genes to transform eukaryotic cells and force them to do what the bacteria or viruses want. So similarly in biotechnology, what do? We force our gene of interest to be inserted into the host cell, okay, and want them to produce the product that we want, right? Like we want to produce maybe insulin. So we are forcing our gene of interest to be inserted into those host cell, okay, maybe a bacteria. And once the gene of interest is inserted into the bacteria, we are making them to do what we want, okay. We want them to produce insulin, and so they are able to produce insulin because we have inserted forcefully a gene of interest into those cells, right? So yeah, pathogens, they are doing this from a long period of time. Viruses, bacteria, they were doing it. Pathogen like Agrobacterium tumefaciens, this is a pathogen for a dicot plant, okay? And this pathogen, it is able to insert a piece of DNA which is called as tDNA. In the last class, we talked about this part, that what do we have? We have Ti plasmid, okay? Agrobacterium tumefaciens. What does it have? It has Ti plasmid. And this Ti plasmid, it is having two regions, right? One of the region is tDNA region, and this tDNA region is detached from this Ti plasmid. This tDNA will be inserted into your plant cell, and then the plant cell, normal innocent plant cell, they will be converted into your cancerous tumor cells. Okay, so yeah, remember this thing, that Agrobacterium tumefaciens, it is a pathogen for several dicot plants, and it is able to deliver the piece of DNA known as tDNA to transform normal plant cells into the tumor cells and direct these tumor cells to produce the chemicals that they want. Okay, so yeah, basically normal plant cells, they are becoming tumor cells and they are producing the chemicals which are utilized by these bacteria. Okay, so yeah, they are making the plant cell to do what they want, what the bacteria want. Okay, similarly, what do we have in the animal cells? To transform the animal cells, we have retroviruses, and retroviruses, once they enter into the host cell, animal cell, they convert proto-oncogene into oncogene and convert normal cell into cancerous cell. Okay, so yeah, retrovirus in animal, they have the ability to transform normal cells into the cancerous cells by delivering their genome into the host cell. Okay, so yes, by understanding their mechanism of infection, scientists, they were able to understand that by using these pathogen only, we can easily deliver our gene of interest. So what do? We just take Agrobacterium tumefaciens, we take retroviruses, we tweak them, we tweak their genome. The TI plasmid that we have in Agrobacterium tumefaciens, we modify it so that instead of having cancer causing gene, they have our gene of interest. And while the tDNA piece is delivered, the tDNA piece is not having the cancerous gene. This tDNA piece is going to have our gene of interest. So Agrobacterium tumefaciens is doing its own task. It is unaware that my TI plasmid is modified. It is just naturally infecting the dicot plant, naturally transferring the tDNA, but little does it know that the tDNA is having our gene of interest because we have modified it. Okay, so yeah, that's the thing. So yes, tumor inducing plasmid is one such vector, example of vector. Okay, TI plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector, which is no more pathogenic. So even if it is delivering tDNA, plant, they are all right. They will not have any cancer, they will not have any tumor. But our gene of interest is transferred into the plants. Okay, so TI plasmid of Agrobacterium tumefaciens, it is modified into cloning vector, which is no more pathogenic to plant, but still able to use the mechanism to deliver the gene of our interest. Similarly, we have also modified the retroviruses, and these retroviruses also has now become non-pathogenic. I'm not saying that all the retroviruses has been modified. Okay, some of the retroviruses which causes cancer, those retroviruses we have modified them in the lab and we are using them to transform the animal cells. Okay, so retroviruses, they also have been disarmed. Disarm means whatever harmful genes they were having, those genes are removed and gene of interest has been inserted. Okay, and now they are being used as a vector to deliver the desirable gene into animal cells. So once a gene or a DNA fragment has been ligated into a suitable vector, we can achieve whatever we want. We want multiple copies of our gene to make multiple products, like we want to make product in a bulk form, so we can use a plasmid which can have multiple high copy number. If we want to transform an animal cell, of course we can use the retrovirus which naturally can just transform your animal cell. If you want to transform a plant cell, you can use TI plasmid of Agrobacterium tumefaciens. If you want to transform your bacterial cell like E. coli, you can use a plasmid which can easily be absorbed by your bacterial cell. So these are the example of your vectors. Okay. So that is it from the NCERT part. Now, very small topic that we are having today, competent host. Okay. So normally what happens? We make recombinant DNA. Okay, we make recombinant plasmid. And then we transfer this recombinant plasmid into the host. But do you think any living cell is so foolish that it is going to take up anything from the surrounding? Any foreign DNA is there in the surrounding, it will be able to take it from the surrounding? No. Your cell, it has Animal cell, it has cell membrane. Bacterial cell, it has cell wall and a cell membrane. Okay. So basically what do we have? If you talk about the cell wall of bacteria, there is no problem with the cell wall because it is freely permeable. Right? It will allow any material to pass through it. No problem with that. But then it also has the cell membrane. It also has the cell membrane. And because the cell membrane is selectively permeable, it is selectively permeable, it will not allow anything to pass through it unless it is useful, unless it is required by your cell. Also, there is one more thing. If you talk about cell membrane, you have learned about this thing in botany, I think, in cell biology, that cell membrane is a bilipid layer. Lipid is polar or non-polar, tell me. Lipid, fats, they are polar or non-polar? They are charged or neutral? Non-polar. Very good. Absolutely correct. Both of you, Abdul and Shafeen. They are non-polar. Absolutely correct, Zuha. They are non-polar. Right. So, bilipid layer of cell membrane, it is non-polar. Tell me about DNA. DNA is neutral or is it charged?
Negatively charged.
Negatively charged. Absolutely correct. Very good, Sumaya and Shafeen. Very good, Mariam. Yes, very good, Fatima. Absolutely correct. So, DNA is a polar molecule. It is having a negative charge, right? So, remember this thing that if you talk about a cell membrane, what do we have? In the cell membrane, we have bilipid layer, right? We have this hydrophilic head region, and this hydrophilic head region is going to have hydrophobic tail. Okay, this is one lipid layer, and similarly, we are also going to have one more lipid layer. So, if we talk about a normal cell, what do we have? Normal cell is having cell membrane, and the cell membrane is having what? A cell membrane is having a bilipid layer. In this bilipid layer, what do we have? We have hydrophilic head, which is polar. No problem with this also. It will allow DNA to pass, okay, because it is hydrophilic. Hydro means water, philic you already might have learnt in chemistry, means love, loving, right? So it is water living molecule. Hydrophilic head part is there. Then we have this small hydrophobic tail region. Okay, so this is something which is non-polar. This is non-polar. And hydrophilic head, it is polar, no problem with this. It will allow, okay. Cell membrane, cell wall, it will allow. But in the cell membrane, the hydrophobic part which is there, it will not allow the DNA molecule to pass through it, okay. The DNA molecule which we are having, this DNA molecule, or let me just write alien DNA. This alien DNA, it is polar, okay, because it is negatively charged. Why the DNA is negatively charged? Why DNA molecules, they have the negative charge?
Because of the phosphate group.
Because of the phosphate group which is present in sugar phosphate backbone of the DNA, right? Very good. So yes, remember this thing. DNA, it is going to be negatively charged because it is having phosphate group present in sugar phosphate backbone. Okay. So this is the problem. Normally your cells which are there, your host cells which are there, they cannot take up any foreign DNA. They cannot take up any alien DNA. So we can say they are non-competent. Okay, they are non-competent to take up our DNA. And that's why there is a requirement to make them competent. Okay, so we have to make them competent so that they can take up alien DNA from the surrounding. Okay, so what do we do? There are two steps that we follow, okay, to make host cell to be competent. So that they absorb, or you can say they can take up alien DNA from the surrounding. We have two things. Number one, we treat the host cell with bivalent cations. Okay, bivalent meaning two valency, cation meaning positive charge. So we treat the cell, host cell, with bivalent cation. And what happens due to this? Positive charge will be accumulated here. And whenever the DNA is going to, the DNA trying to pass through this part, it will somehow kind of balance your DNA molecule. DNA, they are negatively charged, and bivalent cation, they will just accumulate near the cell membrane. So this will ease the process of DNA transfer. Okay, the permeability of DNA will be increased. That is one thing. So yes, we can treat the host cell with bivalent cations. Example, example calcium which is present in calcium chloride. Okay, so yeah, we can use this. We can just easily treat the culture of your clones, bacterial clones, with this particular calcium chloride. Okay, now second thing that we can do after this is to give heat and cold shock to the cell, to the host cell which are there. And due to this heat and cold shock, what will happen? This will create transient pore. Transient means temporary pores. Okay, this will create transient, or you can say temporary pores in the cell membrane of bacteria. Okay, so now what were the problem? The problem is cell membrane, it is freely permeable, selectively permeable, right? Cell membrane, it is selectively permeable, and cell membrane has a part which is non-polar. So to treat the non-polar part, we have treated it with calcium chloride. Okay, and also to make it a bit more permeable, we are creating pores in the cell membrane by giving it some heat and cold shock. Because of the cold shock, cell membrane will contract. Then because of the heat shock, cell membrane will expand. Contraction, expansion, contraction, expansion. Due to this, there will be some pores that will be created. Okay, of course, by the time these pores will be removed, it would be resolved, but for temporary period of time, when we are giving the shock, there will be temporary pores that will be created in the cell membrane. And due to these two reasons, efficiency by which bacteria take up DNA it is going to increase. Okay, initially you were just having your bacterial clones in the culture medium. You were having recombinant plasmids released into the culture medium. So maybe one or two bacteria might take up foreign DNA. Okay, because it is almost impossible for your bacteria to take any foreign DNA from the surrounding. So some of them they will take. But to increase the efficiency by which they absorb the DNA, what do? We treat them with calcium chloride so that cations are accumulated near the cell wall, cell membrane. Okay. This increases the permeability of DNA molecule. Also, we are giving your bacterial cells some heat and cold shock, due to which temporary pores are created, and because temporary pores are there, easily DNA will be able to pass. Temporary pores are created, DNA will be able to pass. Okay, so that's the main thing. Okay, that's how we make the cell competent to take up the DNA. Okay, so remember this thing. Efficiency is going to increase of the bacteria to take up DNA. By these two methods. Okay. So yes, that is it. Do we have time? Ouch. We have. Let me know, guys, if we have any issue. We can talk about the transfer of DNA as well. We have 40 minutes. No, Abdul. We have.
I mean, it's 8:20 right now here.
Oh, I mean, but our class is going to be over in 25 minutes. I just meant to say that. Yeah. So, yes. If it is clear, shall we talk about the transfer of DNA? Everybody, is it done? Have you written down it in your notes? I think Sufi is still writing.
Yes, ma'am.
What about others? Any doubts? Still writing, Zuha? Okay, let me know, Zuha, when you are done. Sufi is done. Nice. I hope everybody is making notes, right? Yes, nice. Done. Okay, great. Let's talk about transfer of DNA. Okay, but okay, I'll show you diagrams in the next class, but I'll just tell you how exactly the DNA is transferred. Okay. So transfer of DNA, it take place by two methods. One is direct method. And the other one is indirect. In the direct method, we are just directly inserting our gene of interest into the host cell, and after being inserted into the host cell, that gene, it is required to be integrated with the host DNA so that it can start its replication. Okay. So here in the direct method, we have two more method like under this category. One is your micro injection, something which is also a part of assisted reproductive technology. Okay. In micro injection method, remember this can be used to transfer the foreign DNA into the plant, into the animal cell. Okay. For example, when we have to, transfer, what do we say? Basically, we have to perform the fertilization process where we are using the micro injection method, and with the help of micro injection, directly the genome of the sperm, it is just injected into the nucleus of the ovum. Okay. So in this micro injection method, what do we use? We use micropipette, and using the micropipette, using the micro injection, sorry, we are just directly injecting the genome into the nucleus of the other cell. Okay, so that the desirable DNA, desirable gene of interest, it is just inserted into the nucleus, and soon it is going to be integrated into the DNA of the host. Okay, then we have one more method which is called as biolistic or gene gun method. And in this gene gun method, we are using a biological gun in which, of course, the particles which are having the DNA molecules, they are loaded, and with the help of gun, we are just directly shooting the particles into the animal cell. So this particular method, it can be used to insert the foreign gene, foreign DNA into the plant cell. In this method, what do we do? The DNA that we are having, these DNA particles, they are coated with some what do we say? Some particles which are non-reactive, some inert particles which are there. Some of the example of inert particle, you guys tell me. Inert metals, inert metals. Gold, gold, yes, Shafeen, good. So we have gold, we have tungsten, and these metals, they are kind of non-reactive, they're kind of inert, right? So of course, these DNA particles, they will be coated with DNA particles, they are coated with inert metal such as gold or tungsten. Okay, and then they are shot gold plated DNA particle we are having, and these are shot using, of course, the biolistic gun or the gene gun. Okay, so they are shot into the plant cell which is there. Okay, specific velocity has to be set so that the particle do not just pass the whole cell and from the other end it goes out. So velocity has to be set so much that it can penetrate the cell wall, cell membrane and the nuclear membrane. Okay, and a bit less, okay, around which it does not escape the whole cell. Okay, so yeah, that is the biolistic or gene gun method. I'm going to show you the diagram so that you are able to understand, but there is nothing much to talk about. For the micro injection, I think I'll use one GIF, and the biolistic gun, this is nothing but just remember the DNA particles, they are coated with some inert metals and then they are shot with the gene gun at the plant cell. Okay, in indirect method, as the name suggests, we are not directly inserting our gene of interest. So what do? The gene of interest, first of all, it is ligated to the vector, and then vector takes up your gene of interest into the host cell. Okay, so here in the case of indirect method, we are using vector. Okay, so gene of interest, it is inserted into vector, and this vector is going to act like a vehicle to transfer our gene of interest. Okay, so this vector will act as a vehicle to transfer the gene of interest. Example of vector, just now we have talked about TI plasmid of Agrobacterium tumefacium. We have retroviruses also that we can use. We have plasmids also of bacteria which can be easily absorbed by bacteria. So these are the examples of vector. Okay, So this is it for the transfer of gene of interest. One we have direct method, other we have indirect method. Okay. Yes. Also remember which method is utilized for transferring the gene of interest directly into the animal cell, which method is preferred for transferring gene of interest directly into the animal cell. Okay, for animal cell microinjection is there, for plant cell bioelastic or gene gun method is there, which will just short the particles at the plant cell, and particle which DNA particles which are coated with gold, tungsten, they will be entering inside the nucleus of plant cell. Okay. Any doubts that we might be having? No doubts? If no doubts are there, I can take your leave. On this Saturday, do we have a booster class? I think we do have. So what do you say, guys? Which topic should I take up for booster class? Any topic that you are feeling like we have not practiced much question? Tell me. Chapter, come on. Which chapter should be taken up in the booster class?
Human health and disease.
Human health and disease. Okay. Okay, done. But I think in the last class, were we having the same chapter? Last booster class. Okay, let's take up human health and diseases only. Okay.
Okay, ma'am.
I cannot read everybody's messages. Can you please send me a full stop? Okay, no, I cannot read it yet. Let me see. I'll just come back. Okay, so human health and disease, only one message I could see of Sufi. All right. How is biology, guys? Huh? Awesome, amazing. Why are you asking, Abdul? What about you? Anyways, any chances that we could, just have the names revealed for, Darwin's ex-wife, then, different names, Albert's wife. I don't know how many names were there. So any chances anybody is going to reveal their name? And yeah, what about the person who was like Jyoti's mom favorite? I want to know. I want to know about that person. Physics Baba for no reason. Once a secret, always a secret. Well, true. Anybody, but there are anyone who is willing to, just tell me the name because I want to know. Arey, a word is bothering me a lot, tunicata. What is it? What was it? Tunicata, this is the word which is used for your protochordates, Abdul. Protochordates because they have the outermost covering, which is, what do we say? In the protochordates also, you have one was, okay. Basically, you have, I just forgot the main thing here. In the protochordate, we have the two category. One was cephalochordate and the other one was, what was it? So basically, tunicata, they were the organism who undergo retrogressive metamorphism, and they lose their, notochord-like structure which was present in the tail. during the metamorphosis, right? Example is your herd mania. What was the category? So basically, yeah, Abdul, herd mania, because they have the covering which is made up of tunicate. They are also called as tunicatas. Okay. No, yes, they are invertebrates, but they are the part of chordates. Okay. Yes. They are invertebrates, but chordates. Okay, so everybody, I think I can take your leave because you guys might be tired, or if you want to talk for 5 minutes, you can talk. Okay. You can have a discussion how your tests or maybe exams are going or anything else. Yeah. Or maybe we can have a discussion on this Abdul doubts. Right. Abdul, is your doubt resolved? You have made me confused. Like, I'll have to check what was the category. Euro coordinate it was. Yes. Oh, somebody also just mentioned it. Yes, Mariam, absolutely correct. It was Euro coordinate. Thank you. Well, people, shall we take the leave now? You can just have a rest. I think we
Yeah.
will just meet in the booster class with the human health and disease chapter only, or we can have a mix questions. Okay, let's have 50 questions this time, and we will have like a rapid fire round. Immediately you have to answer.
Rapid fire. Yes,
Okay,
yes.
so yes, we will have around 50 plus questions from all the topics: human health and disease, human reproduction, human reproductive health, and the evolution chapter. Okay, all 4 chapters, and yeah, 50 plus questions from PYQs.
Okay.
Okay, so I'll see you in the next class, everybody. Till then, just take care. Have a great day. Bye bye. Thank you for joining.
Yeah.
Have a good
Thank
day.
you so much, ma'am. Bye.
Bye bye. Good night.
Good night.